Imagine a river flowing through a complex landscape. This river represents your bloodstream, the lifeline that nourishes every organ, every tissue, and every cell in your body. Now, imagine that the pressure within this river begins to rise. At first, the banks held firm, the landscape adapts. But over time, the relentless force of the rushing water begins to erode the foundation. The pipes strain, the delicate filtration systems become overwhelmed. This is the reality of hypertension, or high blood pressure, a condition often referred to as the silent killer, because it operates without symptoms, quietly dismantling the body's infrastructure from the inside out. For decades, the medical community has battled this condition with a standard arsenal of daily pills. We have diuretics to flush out fluid, beta blockers to slow the heart, and inhibitors to relax the vessels. These tools are effective, but they come with a significant flaw. They rely on the human element. They rely on memory, on routine, and on the fluctuating absorption of a daily tablet. But what if there was a different way? What if instead of treating the symptoms of the pressure daily, we could turn down the pressure at the source? What if we could send a molecular message to the liver, instructing it to stop producing the very protein that sets the chain reaction of high blood pressure in motion? Today, we are diving deep into a fascinating frontier of clinical science. We are looking at a clinical trial that explores a new class of medicine, one that utilizes the body's own genetic mechanisms to silence a specific signal. This is not just a story about a new drug. It is a story about RNA interference, the Renin-Angiotensin-Aldosterone System, and the relentless pursuit of precision medicine. We are looking at the KARDIA-2 study, a piece of research that aims to rewrite the protocol for how we manage one of the world's most common chronic conditions. Welcome back to Clinical Trial Explained. Here we dedicate our time to exploring the intricate, rigorous, and fascinating world of clinical research. Our mission is to take the complex data found in medical registries and scientific protocols and translate it into a narrative that reveals the incredible work happening behind the scenes of modern medicine. Before we embark on today's extensive journey into the science of hypertension and gene silencing, I must provide a critical disclaimer.
The content presented here is strictly for educational and informational purposes. We are analyzing the design, rationale, and scientific background of a specific clinical trial registered under the identifier mentioned in the description. Nothing you hear in this narration should be construed as medical advice, a recommendation for treatment, or a suggestion to change your current healthcare regimen. Clinical trials are experimental by nature. They are designed to ask questions, not to guarantee answers. The safety and efficacy of the investigational agents we discuss are still under scrutiny by regulatory bodies and scientific experts. If you have concerns about your blood pressure or your health, please consult a qualified healthcare professional. With that established, let us settle in. We have a lot of ground to cover. To understand the significance of the KARDIA-2 trial, we cannot simply look at the study design in isolation. We must first understand the enemy it fights, hypertension, and the biological machinery it attempts to dismantle. We must look at the history of how we got here and the microscopic interactions that define this new therapeutic approach. Let us begin our deep dive into the science of Zilebesiran and the future of cardiovascular research. To fully appreciate the gravity of the research we are analyzing today, we must first confront the scale of the problem. Hypertension is not merely a number on a cuff. It is a global health crisis of staggering proportions. It is estimated that over 1 billion people worldwide are living with high blood pressure. That is 1 in every 8 people on this planet. In many populations, as we age, the prevalence rises even higher. It is the leading risk factor for cardiovascular death, contributing to heart attacks, strokes, heart failure, and kidney disease. But why does this matter so much in the context of research? Why are scientists still looking for new treatments when we already have shelves full of antihypertensive medications at every pharmacy? The answer lies in the limitations of our current paradigm. Consider the life of a patient with hypertension. To stay safe, they often must take 1, 2, or even 3 different pills every single day. This regimen requires perfect adherence. However, human beings are not perfect. We forget. We travel and leave prescriptions behind. We encounter side effects that discourage us from continuing therapy.
Furthermore, oral medications have a pharmacokinetic profile that creates peaks and troughs. When you take a pill in the morning, the concentration of the drug in your blood rises, peaking a few hours later, and then slowly declining until the next dose. This means that in the early morning hours, right before the next dose is due, the protection may be at its weakest. Ironically, this early morning period is often when the risk of cardiovascular events like stroke is highest. The research we are discussing today matters because it represents a potential paradigm shift from daily maintenance to durable long-term control. The concept being tested is whether a single injection could provide consistent blood pressure reduction for months at a time. This would eliminate the daily burden of pill taking. It would smooth out the peaks and troughs, potentially offering 24-hour coverage that remains steady over weeks and months. But beyond the convenience, this research matters because it targets the physiology of hypertension with a level of precision that was the stuff of science fiction only a few decades ago. We are moving away from drugs that simply dilate pipes or flush water and moving toward therapies that interact with the genetic instructions of the body itself. This trial, KARDIA-2, is investigating whether adding this new genetic approach to standard background therapies can offer better control for patients whose blood pressure remains uncontrolled despite taking medication. It addresses a critical unmet need: the patient who is taking their standard medication, but still cannot get their numbers down to a safe range. Understanding this trial helps us understand the future of chronic disease management. It highlights a move towards siRNA, or small interfering RNA therapeutics, a modality that could eventually be applied to cholesterol, metabolic diseases, and beyond. Therefore, dissecting this study is not just about blood pressure, it is about witnessing the evolution of pharmacology itself. Now, let us turn our attention to the specific subject of this inquiry. The researchers involved in the KARDIA-2 trial are studying an investigational drug known as Zilebesiran, also referred to in scientific literature as ALN-AGT.
To understand what Zilebesiran is and what it does, we have to take a journey into human physiology, specifically into a complex hormonal cascade known as the Renin-Angiotensin-Aldosterone System, or RAAS for short. The RAAS is the body's master regulator of blood pressure and fluid balance. It is an ancient, evolutionary mechanism designed to keep us alive. In the days of early humanity, the primary threats to survival were dehydration and bleeding. If a hunter-gatherer was injured or could not find water, their blood pressure would drop. The RAAS would kick into gear to clamp down blood vessels and retain salt and water, maintaining pressure to keep the heart and brain perfused. It was a survival system. However, in the modern world, where salt is abundant and physical trauma is less common than chronic stress and obesity, this system often becomes maladaptive. It stays permanently switched on, driving blood pressure up to dangerous levels. Let us trace the pathway that Zilebesiran targets. It begins in the liver. The liver produces a protein called angiotensinogen. Think of angiotensinogen as the raw material, or the precursor. It is an inert protein floating in the bloodstream, waiting for a signal. When the kidney detects low blood pressure or low sodium, it releases an enzyme called renin. Renin acts like a pair of molecular scissors. It finds the angiotensinogen and cuts a piece off. This resulting piece is called angiotensin-1. Angiotensin-1 is still relatively inactive. It flows through the bloodstream until it reaches the lungs and other tissues, where it encounters another enzyme called angiotensin-converting enzyme, or ACE. You may have heard of ACE inhibitors. They work right here. ACE cuts the molecule again, transforming it into angiotensin-2. Angiotensin-2 is the biologically active, potent end product. It is a powerful vasoconstrictor, meaning it causes the muscular walls of the arteries to tighten and narrow. Imagine putting your thumb over the end of a garden hose. The pressure shoots up. Angiotensin-2 also tells the adrenal glands to release aldosterone, which tells the kidneys to keep salt and water in the body, adding more volume to the system. More volume and tighter pipes equals high blood pressure. Most current blood pressure drugs work downstream. ACE inhibitors try to stop the conversion in the lungs. ARBs, or angiotensin receptor blockers, try to stop angiotensin-2 from binding to the vessels. But the liver keeps churning out the raw material, angiotensinogen, regardless of what drugs you take. This is where Zilebesiran enters the picture. Zilebesiran is an RNA interference therapeutic. It does not block the protein after it is made. It stops the protein from being made in the first place.
To explain this, we must look at the central dogma of biology. DNA makes RNA, and RNA makes protein. The instructions for making angiotensinogen are stored in your DNA in the nucleus of your liver cells. When the liver needs to make angiotensinogen, it copies that instruction onto a messenger molecule called messenger RNA, or mRNA. This mRNA travels out of the nucleus to the cellular factories, the ribosomes, which read the recipe and build the protein. Zilebesiran is a small, double-stranded piece of RNA designed to match the specific sequence of the angiotensinogen mRNA. It is conjugated or attached to a specific sugar molecule called GalNAc. This sugar acts like a VIP pass that allows the drug to be absorbed specifically by liver cells. Once inside the liver cell, Zilebesiran loads into a cellular complex called the RISC. This complex finds the natural mRNA for angiotensinogen and slices it in half. Because the mRNA is destroyed, the ribosomes cannot read the recipe. The protein is never built. The levels of angiotensinogen in the blood drop significantly. With less raw material available, the body cannot produce as much angiotensin-2, even if renin levels rise. It effectively turns off the tap at the source. The researchers in the KARDIA-2 study are investigating the efficacy and safety of this mechanism when used in combination with standard oral medications. They are asking, if we have a patient taking a standard drug like olmesartan, amlodipine, or indapamide, and we add Zilebesiran, do we see a greater synergistic reduction in blood pressure compared to adding a placebo? Having established the biological target, we must now meticulously examine the architecture of the trial itself. A clinical trial protocol is a blueprint for scientific discovery, ensuring that the data collected is unbiased, robust, and meaningful. The study we are discussing is a Phase 2, randomized, double-blind, placebo-controlled, multi-center study. Let us unpack each of those terms to understand the rigor involved. Phase 2 implies that this drug has already passed initial safety testing in a smaller group of humans, Phase 1.
Now, the goal is to determine the optimal dose and get a clearer picture of efficacy in the target patient population. Randomized means that patients are assigned to treatment groups by chance, like the flip of a coin, to ensure the groups are comparable. Double-blind is the gold standard of research. It means neither the patients nor the doctors know who was receiving the active drug and who was receiving the placebo. This prevents the power of suggestion or researcher bias from influencing the results. The study population focuses on adults with mild to moderate hypertension. These are individuals whose systolic blood pressure, the top number, is between 130 and 160 millimeters of mercury, despite being untreated or washed out of medication. The trial is structured into several distinct periods, each serving a crucial scientific function. First, there is the screening period. Potential participants undergo rigorous testing to ensure they meet all criteria. They must have confirmed hypertension, but cannot have severe cardiovascular instability that would make participation unsafe. Next comes the run-in period. This is a vital phase where the researchers standardize the baseline. Patients stop taking their previous blood pressure medications. Instead, they are started on a specific open-label background therapy. In this study, there are 3 distinct cohorts, or groups, based on the background medication provided. Cohort 1 receives olmesartan. Olmesartan is an angiotensin receptor blocker, or ARB. It works by blocking the receptor where angiotensin-2 tries to dock. Cohort 2 receives amlodipine. Amlodipine is a calcium channel blocker. It prevents calcium from entering the muscle cells of the heart and arteries, allowing them to relax and widen. Cohort 3 receives indapamide. Indapamide is a diuretic. It helps the kidneys filter out excess Alton water, reducing the volume of fluid in the blood vessels. This run-in period lasts for at least 4 weeks. The purpose is to ensure that every patient is stable on one of these standard drugs before the experimental drug is introduced. It creates a clean scientific slate. After the run-in period, patients who adhere to the daily pill and still meet the blood pressure criteria enter the randomization phase. This is day 1.
Within each of the 3 cohorts, patients are randomly assigned to receive a single subcutaneous injection of either Zilebesiran, the active drug, or a placebo, a saline solution with no active ingredients. The dose of Zilebesiran used in the specific protocol is 600 milligrams. The injection is given under the skin, typically in the abdomen, thigh, or upper arm. Following this single injection, the double-blind treatment period begins. This period lasts for 6 months. During this time, patients continue to take their daily background pill, olmesartan, amlodipine, or indapamide, every single day. They visit the clinic regularly for blood pressure checks, blood draws, and safety assessments. The primary endpoint, or the main question the study asks, is the change in systolic blood pressure from baseline to month 3. The researchers measure the blood pressure using 24-hour ambulatory blood pressure monitoring, or ABPM. This involves the patient wearing a cuff that automatically inflates every 15 to 30 minutes for a full day and night. ABPM is considered the most accurate way to measure blood pressure because it captures the true average, avoiding the white coat effect, where anxiety at the doctor's office spikes readings. The researchers are looking for a statistically significant difference. Does the group that got the Zilebesiran shot plus the daily pill have a lower blood pressure at month 3 compared to the group that got the placebo shot plus the daily pill? Secondary endpoints look at the durability of the effect. They measure blood pressure at month 6 to see if the single injection is still working. They also look at biomarkers, specifically the levels of serum angiotensinogen. They expect to see these levels plummet in the treated group, confirming the drug is doing its job of silencing the gene. The study design essentially asks, can we supercharge the effect of standard pills with this genetic silence? Does combining an upstream inhibitor, Zilebesiran stopping production, with a downstream blocker, olmesartan or amlodipine handling the rest, provide superior control? And importantly, does it do so safely? As we contemplate the architecture of the KARDIA-2 study, we must also allow ourselves to look forward to the potential implications of this research. What does it mean for the future of medicine if this approach proves successful? First and foremost, it addresses the issue of therapeutic inertia.
Often, doctors are hesitant to add a second or third pill to a patient's regimen because they worry about complexity and side effects. A twice-yearly injection that essentially vaccinates the patient against high angiotensinogen levels could simplify treatment protocols immensely. A patient could visit their cardiologist, receive their injection, and know that they have a foundational level of blood pressure control that will not wear off if they miss a pill one morning. Secondly, this research explores the concept of consistent 24-hour control. As we discussed earlier, oral medications have half-lives. Their effects wane. By suppressing the production of the protein at the liver, Zilebesiran theoretically provides a flat, steady line of suppression. There are no peaks or troughs. This stability is crucial because blood pressure variability, the swinging of pressure up and down, is an independent risk factor for organ damage. A steady pressure is gentler on the kidneys, the eyes, and the brain. Furthermore, this study helps researchers understand combination physiology. By testing Zilebesiran against 3 different mechanisms, an ARB, a calcium channel blocker, and a diuretic, scientists can learn which combination yields the best results. For example, perhaps silencing the liver's production works best when paired with a diuretic that removes sodium, or perhaps it works best with a calcium channel blocker that relaxes the vessels. These insights allow for personalized medicine, where a treatment plan is tailored to the specific physiological needs of the patient. It is also worth noting the broader implication for RNA interference technology. If Zilebesiran is successful and safe in a population as large and diverse as the hypertension community, it validates the platform for other common diseases. It suggests that we can move from treating symptoms to silencing disease drivers at the genetic level for chronic conditions that affect millions, not just rare genetic disorders. However, we must temper this optimism with scientific caution. A successful phase 2 trial is a milestone, not the finish line. It tells us the drug works in a controlled setting. It paves the way for phase 3, which involves thousands of patients and looks for rarer side effects. But the potential to transform a daily burden into a biannual procedure is a powerful motivator for this continued inquiry.
No discussion of a clinical trial is complete without a rigorous examination of safety and ethics. In fact, in the hierarchy of clinical research, safety always sits above efficacy. A drug that works perfectly but causes unacceptable harm is of no use to medicine. In the KARDIA-2 trial, the safety monitoring is extensive. Because Zilebesiran targets the kidney's regulation system, the RAAS, researchers must be hypervigilant about kidney function. They monitor creatinine and potassium levels. The RAAS helps the kidneys excrete potassium. If you block the system too aggressively, potassium levels can rise, leading to hyperkalemia, which can cause dangerous heart rhythms. This is why the exclusion criteria for the trial likely prevent patients with severe pre-existing kidney disease from joining. Another critical safety concern is hypotension, or low blood pressure. While the goal is to lower pressure, lowering it too much or too quickly can cause fainting, dizziness, and falls. Since Zilebesiran is a long-acting injection, you cannot simply stop taking it if the pressure gets too low, as you would with a daily pill. Once injected, the drug is active for months. Therefore, the trial includes strict rescue protocols and monitoring to manage any instances where blood pressure might drop too low. Researchers also monitor for injection site reactions. Since this is a subcutaneous shot, they look for redness, swelling, or pain at the site of administration. They also monitor liver enzymes. Since the drug targets liver cells and accumulates there, it is vital to ensure it does not cause inflammation or toxicity to the liver itself. Ethically, the use of a placebo in this trial is justified because no patient is left completely untreated. All participants, regardless of whether they get the active injection or the placebo injection, continue to receive a standard of care daily medication, olmesartan, amlodipine, or indapamide. This ensures that everyone is receiving a baseline level of therapy for their condition. It would be unethical to give a hypertensive patient a placebo alone without any background support, but adding a placebo to a standard therapy is a recognized and ethical way to test a new add-on treatment. Informed consent is the bedrock of this ethical framework. Every participant in the KARDIA-2 study must be fully educated on the potential risks, the experimental nature of the drug, and their right to withdraw at any time.
They are partners in the research, not just subjects. The data derived from their participation helps build the safety profile that regulatory agencies like the FDA or EMA will eventually review to decide if this drug should be approved for the general public.